Dr. Butterfield is a clinical scientist focused on transitional research in neuromuscular disorders. As I mentioned earlier, he's currently an associate professor in the departments of pediatric and neurology at the University of Utah, where he directs the Primary Children's Hospital and the Shriners Hospital in Salt Lake City. His primary emphasis is focused on genetic therapies for neuromuscular disorders and understanding genetic modifiers of severity in various types of neuromuscular disorders. So thank you so much, Dr. Butterfield, for being here. As you can tell, he's a very, very busy man and he has a lot of patience. So I appreciate your time. And welcome to the July Early-onset Chapter meeting.
So just to maybe give a little more background on myself, I'm a pediatrician at heart. So I was interested when I heard about this early onset group with FSHD. So I think this is a really nice group and a good way to fill some gaps within the community that are largely adult focused, adult FSHD focused. So this is a good thing. So my background is also in genetics. And so this is not like a formal lecture, right? But I wanted to just maybe, and maybe to give a little background, I think we're coming into an age of genetic medicine, right? And so I think it's important to understand a little bit the background of how we got here, because I think, especially as we look at studies for FSDs that are coming, they're really focused genetic therapies, like how we're treating a gene. But we have all this other jargon around that I don't think always is that well understood. So I'm going to give you my take on some of it. And the first is genomics, because you hear everything has an omics. Now we have proteomics, genomics, blah, blah, blah. They actually, that word actually goes back to a bar at Alta, Utah a few years ago, and somebody just made it up as a way to talk about all the genes. But basically genomics means all the genes, right? How we can use that to help health outcomes. And it's sort of like all these ones and zeros, like it's just the functional equivalent of the massive technological improvements we made in digital technology, right? You can see how A, C, T, and G would go pretty nicely with that digital stuff. But genetic medicine, that's genomic medicine, right? How do we use all that digital, digitized genetic sequence data to like decide something about our health? And we have some studies here in Utah on that subject, but how is genetic medicine different? And to me, that's like, how do I treat one gene in one patient? And that gets at some different nuances of kind of the way we think about all this new genetic technology. So I did a lecture for a bunch of doctors last year, and I was using jargon I thought they would know. And one of them just raised their hand and was like, what's an exon, which is just a basic function. I'll tell you what it is in a minute. But I was like, oh, maybe we should go back to the start. So this is my attempt to go way back to the start on genetics, right? Back to just what is DNA. And DNA is like the primary contents of the nucleus of a cell. And it's the sequence, right? And it's composed of 4 different like letters in a code, A, C, T, and G. And those 4 letters make up the entire alphabet of the genetic code. And you're used to seeing this sort of triple helix looking thing. That's what the DNA looks like. So that contains all the code for all the proteins and all the things you need to make for every cell in your body to do all the things. And every time a cell divides, you have to make a new copy, right? And that process is prone to error. And so we get genetic mutations, or as we kind of have changed in the terminology, variants. And a variant could be anything from a mutation on a gene that causes disease to a genetic change on a gene that, makes you have blue eyes versus green eyes versus, whatever the trait. And that DNA is packaged onto 23 chromosomes, and you have 2 copies of every chromosome, right? So you have 1 from each of your parents. And those, if you take all the letters of that genetic code, there's about 3 billion letters, A, C, T, and G. And so this is kind of what the chromosomes look like if you looked at them under a microscope. And just sort of gee whiz fact, the chromosomes are numbered in order of size. So number 1 is the biggest, and you go all the way up to number 23, and these are little itty bitty chromosomes. This is the chromosome we care about, number 4. And the part we care about is way out here on the very tippy tip on the end, for so-called 4q. So the long arm of that chromosome is called 4q. In this case, this patient has 3 chromosomes 21, and that means they have trisomy 21 or Down syndrome. So as you think about genetics and its function in disease, any error in the sequence can relate in disease, right? And we have, especially if it's in a gene. And in genes, we have about 20,000 of them. But so they're actually a really small part of the DNA. Only about maybe 15% of the code, or even less, is in a gene. All the rest of the sequence in between the genes, we don't really understand it that well. But if you look within a gene, there's two parts. There are exons and introns. These are those things that the doctors didn't even know. I'll show you a picture in a minute. The exons are the part of the genes that make the protein. They're the important part. So when we do genetic sequencing for like some disease I don't know anything about, we sequence the exons. That's one of the ways to do it, because most likely you're going to find what you want. But then there's all these other parts in between the parts of the genes, even within a gene. So we'll come back to that. So this is the other thing I wanted to just remind you, because actually relevant to FSHD is the Human Genome Project. And this goes back to like the mid-1980s, like those days when they were at a bar making up the word genome. And they started in 1990. They spent about $3 billion with multiple government collaborations to get the first draft sequence. And that we just passed the 20-year anniversary of that last year. And they finally made sort of a so-called complete genome in 2003. And they actually were pretty good at accomplishing their goals, which were to identify the genes and all the sequence, put it in databases that are publicly accessible, and make tools for people to analyze it. So it's been really massively successful. We only had that now for about 20 years. And this is where we were, just to show you at the time we got that, like in the clinic. If I wanted to do gene testing to find a mutation in a disease that like showed up at my clinic, I had a karyotype, which is that picture of the chromosomes I showed you a minute ago. And like we could see like if there's an extra chromosome or some big chunk that was missing. We had like metabolic screening for things like PKU that were newborns. And we had PCR testing, which is a way you can sequence like one gene at a time. And Sanger sequencing is the way we did that. And this is like an old gel from an old sequencing gel. And if you look, it has one column for each letter, and you read the gel going up. And you can see how tedious this would be. Like the way I would read this sequence is like C, and then T, and then T, and then probably C, but they get kind of close together, it's getting harder. T, A, G, G, A. And we can read the whole sequence off of that gel. And that's pretty hard. And you could do a smallish gene that way. So that's kind of where we were. And then we made these really rapid advances that allowed the final completion of that sequencing. And we got a whole bunch of new tools. And this is one of them. So now instead of reading off this old gel, the black dots, I read like fluorescent markers. And wow, it's a lot easier to read. And I can actually train the computer how to read that sequence, right? And then this is what made the whole genome possible. This is like a long read sequence. And you can see it's just a bunch of mumbo jumbo computer jargon, right? Except you look right here, there's about 150 bases of code and then a bunch of more computer mumbo jumbo and some other code. So these are codes for the quality of the read, like how confident I am that that position is an A, for example. And this let us use a computer to take these short reads. They're only about 150 letters long. And so we fragment the DNA all apart. We put it in to a sequencer, we get these sequences, and then we use a computer to map them all back together again, right? And that was a really good way. And that's what led us to that whole genome. And now we can do this like routinely in the clinic. Like if you come into my clinic, I have lots of different tools that use this kind of sequencing technology to find out mutation in whatever gene. And in fact, sometimes the very first test we do is a genetic test nowadays. And we even have genetic testing that is, what if I didn't know exactly which gene to look for? Maybe I'll screen for like 100 or 200 genes that I know are involved in muscle disease. Or if I didn't even know that, maybe I'll just screen all 20,000 genes, right? We can do that pretty easily. And that's becoming more routine in the clinics. And now even we have, in the last 20 years, approval of genetic treatments. So for Duchenne muscular dystrophy and SMA, we have treatments that came about in just the last few years, right? And I think I'm running off an old version of these slides that I added. I'm gonna just pop out for a second because there was a really cool slide I added that's not here. Okay, I'm gonna close out this slides for a second and I'm gonna update because I worked on this late last night on a different computer, and I'm going to try to update it. Let's just see if that works. So anyway, if people have questions too while I'm pausing, that'll be fine. Oh yeah, here's the new one. Okay. So anyway, we have these new therapies, right? And like in SMA, we've been using gene therapy now for 3 years. We've been using some genetic manipulation therapies for 5 years. Same with Duchenne. And these are coming for FSHD too, and probably every other genetic disorder. So I'm going to fill you in on a little secret that we didn't know when we finished that human genome back in 2001. That wasn't really finished. And this is the cover of Science, Journal Science from March of this year, where we finally published the whole genome, this time like for real. And if you look at this image that I put up here, this is the chromosomes again. Remember they go in order of size. So chromosome
You didn't
1.
share your slides again, Russ.
Oh. Probably should do that, huh? Thanks for just correcting me there. All right, can you see that? Can you see that?
Yes, I can.
Awesome. So anyway, the chromosomes are the longest ones first. And everything in red on these chromosomes 1 to 23 is sequence that we actually didn't have with that very first whole genome. It's actually kind of a lot. And these are all like repetitive sequences that are really hard to put back together when you're only sequencing a small chunk. And really important among these is down here, the very tip of chromosome 4 that you can barely see. That's where FSHD lives. And that whole part of the human genome was not very well mapped until like 4 months ago. And so one of the tools that came out of this recent flurry of papers on the now real for real whole genome is actually mention of the locus where FSHD is out on chromosome 4. They actually used that as a good example of a part of the genome that was really hard to do, but now they can do. This is the technology they used to do it, and it's technology that we're using in the lab. And I'll show you just a couple pictures of it. It's called a nanopore. And instead of like fragmenting the DNA up into little bits, you can sequence really long chunks of DNA. And you just feed them through a microscopic pore on a device. You can see what this guy's holding in his hand. It's like a little bigger than a thumb drive. And you can sequence a whole genome on there, except instead of those short reads that are like 150 letters long, you can do it in reads that are like thousands of letters long. And for us, we can do up to 40 or 50 or 60,000 at a time. So then we can really get our handle on these weirdly repetitive parts of DNA. Okay. So one more background slide, and this is what we call the central dogma, which is weirdly named, but this is just like information flow in genes, right? And so I wanted to just put this in your head as like a way to think about if you had a genetic therapy, where would you put it? But to know that, you have to know the central dogma. And then the central dogma is here. So you start on this top line. This is a strand of DNA. That's a gene. It has a promoter. That's what makes it start recognizing that there's a gene there. It has this thing called an ATG. That's a start site for where the code for the protein is at. It has these parts marked in black that are introns. They're just parts of code that don't matter. And so we make a long copy of that called RNA, and we cut out all the parts in black, those introns. So there's a splicing process. And we have genetic therapies that alter how gene splicing works. And we have two different, drugs for Duchenne and SMA that both do this actually. And so these are drugs like now FDA approved. We're sort of manipulating the processing of this step from DNA to RNA. Then we take that strand of RNA and we translate it into protein, right? We use the genetic code and we build a protein out. And there's ways we can manipulate that process too, right? All the way out till we get an active protein. So this sort of process you think about like FSHD, where would we try to fit a treatment in FSHD? And or we just replace the gene altogether, right? That's a whole other kettle of fish. But for FSHD, there's one other important part. And so I put in this slide. This is the one I wanted to make sure was in here, actually. It's called epigenetics. So epigenetics is everything else that's not the sequence. And if you think about, remember that picture of the chromosomes I showed, and they're all these tight sort of globby thing that you can see. Well, in the cell, DNA doesn't look like that. As it getting ready to divide, it looks like that. But most of the time, it's sort of wrapped up along these bits of DNA called histones. And the way it wraps up makes a difference in whether you can turn genes on or off. So epigenetics is sort of a way to control whether a gene is turned on or turned off. And that has to do with how it wraps up around these histones and how it kind of winds up on itself. It also has to do with this little yellow tag you see on this gene that's called a methyl group. And so when we're going to talk about methylation with FSHD, and methylation kind of helps predict whether the DNA is wrapped up on those histones and can be turned on, or it can't be turned on. And now we think of potential therapy mechanisms that are here too, not just in the central dogma of the information flow for genes, but on this epigenetic thing too. So does that all make sense?
I have a question for you, Dr. Butterfield, and I think I sort of understand it, but every time I think I sort of understand it, I don't. But what I understand about methylation is a lot of people have done the spit test with Dr. Jones, and that measures methylation. And the way I understand it is, so typically, not always, but typically the number of deletions you have coupled with your methylation percentage. So for instance, I'll just use my son, for example. He has a 5 repeat, but his methylation is only like 5.4%, which is really low. And from what Dr. Jones and other people I've talked to said, the reason why he's so severe is because with a 5 repeat, he shouldn't be that severe, but coupled with the low methylation, that sort of makes him more severe. And I know that a lot of people in the community ask a lot of questions about, I have a 2 repeat, but I'm not that severe, or I have an 8 and I'm very severe. So it's working both hand in hand with the genetics and the epigenetics. So if you have a high methylation and a low repeat, maybe you won't be that severe, or vice versa. Is that correct?
Yeah, and we don't know completely how to predict that, right? And I'll cover that in a couple of slides too, like the methylation bit and what it looks like and how to think about it. Because I think this is the thing I think is so hard to understand FSHD genetics. And like, what does that methylation test from Peter mean? And we're doing some methylation tests that I can show you a little bit about too. To really understand why methylation matters, and I'll just give you a quick background on FSHD kind of more generally and then more specific on the methylation. But just generally speaking, FSHD is dominant. And that means you only need a mutation on one of the 2 copies of that gene, right? And you can pass it to half of your offspring. And the way to sort of build this gene model in your mind is it's caused by something, and this is just for vocabulary, I'll show a picture in a second, a D4Z4 repeat. It's a long, repetitive strand of DNA, and it's called a macrosatellite, is what those structures are called. And for this case, it's like 3,000 letters or bases long, right? And it's way down here at the very tip of chromosome 4 in an area that's just kind of a weird region right at the very end. And most people have like more than 10 repeats of that D4Z4 repeat, but people with FSHD have a smaller number, usually between 1 and 10. And that number, like Ali said, can kind of predict severity to some extent, but not perfectly. And that long repeat is really hard to study. So for many years, like it's so long that you just can't sequence the whole thing in any reasonable way. And then there's an almost exact copy of it on the end of chromosome 10. So anytime we try to like amplify the DNA in the test tube, it amplifies chromosome 10 too, and it's really hard to differentiate. So that D4Z4 is just kind of a puzzler that's been really difficult. And inside that D4Z4 is a gene. and that gene's called DUX4. So hopefully just remember that the vocabulary part, D4, Z4 is the repeat, and DUX4 is the gene. And the role of DUX4 is to turn other genes on and off in development, right? And here's just a picture we made of it. So this is like all 3,000 letters long of it, right? And here's the DUX4 in blue, that's the gene. And there's a couple other sites that we use for like measuring methylation and other things. You don't have to remember all that, but just, we always make it look like a triangle. I don't know who started that tradition in FSHD, but whenever you see models of FSHD and like the clinical papers and stuff, it's always a triangle. Every repeat is a triangle. And this is sort of the idea, and this is what Ali was getting at. So contraction of that array. So if you have more than 10 copies, it's kind of normal. But if you go less than 10, now you have FSHD, and the severity ties to how many copies, right? And what happened, I've got another bit of this picture, right? So here, this person has 1, 2, 3, 4, 5, 6 repeats, and that what happens, and they drew this into this figure. In black is a spot on the DNA that has one of those methyl groups. Remember that epigenetic, that epigenetic kind of control. And if that methylation tag is there, that DNA winds up really tight, and this DUX4 gene just can never get turned on unless we make less repeats, and then you lose the methylation, right? So that's represented here in yellow. So that methylation disappears, and now the gene can, that epigenetic stuff, that wrapping can open up, and we can turn that gene on. And so the whole problem with FSHD is that we have a gene that's not supposed to be turned on, that DUX4 gene, and it gets turned on. And then it kind of causes havoc in the muscle. And there's some really interesting studies that have been done recently that because so now if we turn on a muscle, it causes FSHD. What does it do when it's like supposed to be turned on? Well, it only ever gets turned on when you're like a 4-cell embryo, right? That's you, the 4 little cells, and it stays on for like a few hours, and it turns on a bunch of other genes, and then it turns off forever, and it never ever gets turned on again unless you have FSHD. So it's kind of a crazy mechanism, and it's not like any other genetic mechanism. Like for Duchenne muscular dystrophy, maybe you've got a single letter difference from the normal gene, or you're missing a small part. It's just pretty simple mechanism there. But for this, it's like wildly complicated. And just for the other thing to clarify, FSHD2 is this sort of other type of FSHD where you can have a normal number of repeats, but for reasons other than the contraction, you lose the methylation and the gene gets turned on. And that can be due to other genes. And so, Ali, this gets to your question. So what makes someone more or less severe, right? Is it the amount of methylation on that gene? Is it the number of repeats? Is it some combination? And what we know already is that even, and here in Utah, we study this very big pioneer family that goes back like 8 generations. They all have 6 repeats, and they all have exactly the same genetics, right? But they're even pretty variable amongst themselves, even from siblings that are related to each other. Almost half their DNA is related to each other, and they can be very distinct. So there's other things that are controlling that severity. In our lab, we think that might be other genes, right? Like the FSH22 gene is a good example of a gene that we would think of as a modifier that's like making it worse. We know from FSHD2 though that like those patients have very low methylation, but it doesn't necessarily correlate with very much more severe disease, right? For reasons we don't know. So it's not completely predictable either on size or on methylation alone. And what Peter's measuring is sort of an average methylation across this very last repeat, this one down here. So when he gives you that number, that's sort of what that's representing. I'm just going to show you here. This is just a small bit of work that we're doing in our lab using that nanopore technique. And this is this pioneer family. So I'm showing you some of the sequencing we've done. And for this technique, instead of just measuring small chunks, we can actually measure the methylation across the whole thing. So so these guys have 6 repeats. It's about 30 kilobases long, and you're seeing the whole thing. And you'd have to zoom way in, but every row in these things is one DNA strand. And if it's marked in red, it has methylation. If it's marked in tan, it has no methylation. And so you can see this patterns in methylation. And I think for our purposes, just know there's patterns there, there's structure there. And so we think that it actually might matter where the change in methylation is that could predict what's going on, right? Does that make sense? So,
So you're saying not only methylation percentage, but on which strand or which repeat has the methylation could also predict severity. Is that what you're saying?
Or just the overall like variability of the methylation. So if you go back, you can see even within this patient number 6-435, some of his strands, some of his chromosome, and think of it maybe nucleus by nucleus. Some of his muscle nuclei have high methylation and some have low methylation. And there's sort of a range within each person. And for these people who all have the same mutation, it seems pretty uniform actually. But we may be able to start now predicting, chromosome to chromosome, or even like within this, right? You can see like a band of methylation here and here and here and here, and it matters where those are. So they're structured inside the DUX4 repeat of methylation too.
Dr. Butterfield, do you know of anything that helps shore up methylation? Like I hear a lot of things like creatine might help, some other stuff might help, folic acid might help shore up methylation. Have you heard anything like that?
I don't know of anything that shores up methylation. It's not for reals, right? There's probably some people who think of stuff. I'm going to just pull up another slide that, John, since you asked, like, what are we doing with your sample? I'm going to show you. This is from data we presented at the IRC a couple weeks ago. But just to say, like, if people know Epic Bio, that's one of the companies that's now working at FSHD. In fact, I saw they just had a press release, it was this week or late last week, about getting some funding. But the way their methodology works is targeting methylation. And so they use a sort of a CRISPR. I know people have heard of CRISPR, but I don't have slides on it. Basically, they use something like a CRISPR to take an enzyme right to the FSHD gene and add methylation on top of it to maybe turn the gene on. But that's sort of the whole idea of what Epic Bio is doing. So John, let me pull this slide up so you can see it. And I got to get pictures out of the way so I can see it. So John, this might be your DNA right here. And this is a little methylation test we developed. So what you're seeing is
It hasn't switched over, right?
Did it not? I'm going to stop share then and restart. Because I can see it and it still says I'm sharing. How's that? You can see the new one?
It's loading. Yes, we're good.
Okay. So, and this is just a slide which and it's kind of technical. This is a tool we use to just measure the methylation in one of the D4 or Z4 repeats, but they're all kind of mixed up. We don't know if it's the first one or the last one, right? And then we see like this graph here. And what this says is we take all the methylation spots on one D4, Z4, and we add them up. What percentage of them were methylated, right? And each one of these dots is a percent methylation in one strand of DNA, one D4, Z4 read, we call it. And you can see the range. So in this unaffected person, they kind of average about 75% on chromosome 10 and a little more than that on chromosome 4, which is where the gene for FSHD lives. Here in this other unaffected person, the chromosome 10 is a little less than 75, and then right about 75 for chromosome 4. But I think this is me. And so John, you and I are on here together. And John, this is an FSHD2 patient. And what's really unique that we found with FSHD2 methylation is that you're low for both chromosome 4 and chromosome 10, and actually quite low. So here you see it's probably about 15% for both chromosome 4 and chromosome 10. And now here's what it looks like in a typical FSHD1 patient. So we see low, it's lower if you have shorter numbers. So this person has a 10 repeat chromosome 10, It's a little lower than the ones I showed you over here. Because this is an FSHD patient now, just with a shorter 10, but this is their chromosome 4. And you see two peaks, right? One is around the higher methylated peaks and one at the lower. So this is their chromosome 4 that's causing the disease. And now here, this patient has two different types of chromosome 4, so we can split them apart. And we see the high methylation in one one of the chromosome 4s and the low methylation in the other one. But the chromosome 10 is normal, right? So one thing that really differentiates FSHD2 from FSHD1 is the low methylation also on chromosome 10. So that's a little extra. John, does that make sense? Since I know that was something you were interested in. And I'm going to stop that share and go back to the other thing. But hopefully so that's just some work we've been doing. We're really interested in the methylation and now have a tool that helps us to really measure it with precision, right? So how we can get back at questions like what you asked. Can you really correlate the level of methylation to the severity? And you just have to have a better tool to measure it, which that's one of the tools we're developing. And then we need better phenotyping. And we're going to talk about phenotyping and natural history kind of next, because this gets at where we can really move things forward. But and that's sort of the end of the genetics bit. Do people have questions or anything? I'm going to reshare here back on this other slide deck. Hopefully you can see that now. Can people see? So this is just a slide I made up last year because I was curious, this table or this figure of like how much is getting published in FSHD right now. So what's happening? And so I just went in PubMed, which is a giant database of all research articles that goes back like 100 years. And I searched on FSHD. That's the only thing I put in the search. And I got these numbers. So every year, like there was one paper in like 1915 and one paper here like 1930. There's this one in 1950 that we really care about. That's the Utah pioneer family. But then look, around 1980, things really started to pick up for about 15 years or so, and then just continued to accelerate. And you just got this exponential curve in the number of publications about FSHD, right? And this all kind of started off in the 80s. And what was going on back then is we developed new genetic tools to map a genetic disease to a location on a chromosome. And FSHD was mapped like the early 1990s to chromosome 4. And that's kind of what represents this big flurry of publication that just kept going. And you see this other chart I made with DUX4, or that gene, right? And remember, that's pretty new. So about 2010 was when that sort of model for genetics of FSHD was published that really implicated DUX4, and you see like just another exponential curve that's really going up like super fast. This is just the number of publications about DUX4, right? So the work in FSHD, if you just go back these last few years, is just going bananas with the number of people interested in and studying FSHD.
So Dr. Butterfield, can I ask you a question? And I don't know if you can answer this off the top of your head. Out of all those research papers, how many are targeting just early onset in kids?
I don't know the number. It's not very many. 5, 10, right? Maybe not 10. Probably not 10. Right? Do you think you could find 10? You might be able to find 5.
5, maybe. And I know that Dr. Katz is trying to do a comprehensive research paper on just early onset with pediatrics, and she's using the U of R registry. Have you spoken with her about that?
I have talked to Dr. Tawil briefly, but not to her. So that's one area where she and I need to just connect. And these papers are not just like clinical papers, right? They're clinical and like researchy things, like someone's interested in some weird genetic mechanism that DUX4. But if you look at all the clinical papers piled together, the early onset would be just a small, a fraction, like a very small fraction, right? So we got a lot of work to do, and I just put up this little list of things, I think, just to put a framework around like making progress in the future, like, because where are the gaps? Like, if we wanted to have a treatment for FSHD, where are the research gaps? Because now we got all this momentum for research, and one of the gaps is that the genetics is really, really hard, right? Like all the tools we had over all of the time going back many years were really hard. And so Peter Jones developed his methylation assay just in the last few years, right? And we developed our nanopore like this year. So, and then we had this publication back in March of the whole genome for, especially for repetitive sequence. And that just happened this year. So we're really actually starting to get at this. Genetics is really hard bit and have tools to get around that. Another problem that makes FSHD is hard that doesn't exist in animals. And when we make an animal model, it's really super engineered. So it's really hard to get like just a natural model in an animal. And that makes it hard to study. And the third thing is we just don't know a lot about the natural history. And by natural history, I mean, how does a disease progress just when we're not treating it in some way, right? Like what happens in that sort of onset, progression, severity of disease in a normal person with this disorder. And we're starting to see studies of this come out, but very few in the early onset group. And I think, and you guys could probably speak to this as much as me, but I think there's been some stigma about diagnosis that has made it hard for some people to engage with the research. You don't want to label your family or insurance or employment issues or concern with FSHD. And the slow progression of disease maybe can make it feel a little less urgent. And that's certainly not the case for particularly an early onset group, right? So there's just been this kind of slowness to move the ball forward. And this is, I think, one of the things that's really moving it forward now is this clinical research or research network or CTRN for FSHD. And this is now, I think, 12 or 13 even centers, including University of Utah, but really centered by Jeff Statlin at Kansas and Robbie Twill at University of Rochester. And the goal is to really start addressing those gaps on the clinical side, right? If you go back to that list of gaps, the genetics is hard, and that's like an engineering problem. We'll figure that out, right? And the FSHD doesn't exist in animals, also an engineering problem. We'll figure that out. Lots of really smart people working on it. But this natural history and the clinical part, like that's where we come in, like where like, I'm a clinician, I work with patients, and like this is where we can make a difference to start to filling that gap, right? And so to do that, we really have to know what is the natural history of disease, right? And that's not easy to do. Those studies take a long time, and we have to know what to measure, right? And really at the end of the day, the question is, if I had a treatment, how would I know if it's working? Like, what would I measure to tell me it's working? And something that I could measure in like a year, not that I would have to wait 10 years to find out, right? Because nobody can do a clinical trial in 10 years. We got to do them in like 1 year. And the Clinical Trials Research Network is really set up to start answering some of those questions. And the first really important trial, I'm not going to tell you about all the trials for everything, but the first really important one is a trial called Resolve. And that trial, it's really just getting kind of wrapped up for most of the patients. And Resolve is an acronym. Those guys at Kansas and Rochester really love acronyms. And you can try to figure out how they got Resolve out of clinical trial readiness to solve barriers to drug development, FSHD. But this is like 300 patients, almost 300 patients who were followed over 2 years. And they measured this long list of things, like at pretty much everything you could think of to measure as like a motor outcome, or even like this test called electrical impedance myography, is a little electrical test on the muscle, or like mouth muscle strength, or DEXA scans, or even just measuring like by hand, like how strong the muscles are, and then a couple other things. And we have DNA samples and plasma samples for other studies. So that study's been going on for a while, and really not much of an early onset platform at all for this study. And it's just kind of getting done. And then the next study that's going to happen is a really important study that we think will have a lot of hopefully interest among the early onset group. And this one's called MOVE. A little easier to pick out the acronym here: Motor Outcomes to Validate Evaluations in FSHD. But the hypothesis here is that there are probably some simple motor function measurements that we can use to predict progression and use those as part of clinical trials, but also use them as a part of like clinical care. Like, how would I know, what to expect in the next year, or a couple of years for a patient that I'm seeing in clinic? And really, as you think about MOVE, it's like an extension out of Resolve. So hopefully we'll enroll a lot of Resolve patients on MOVE. And it's a much smaller list of motor function assessments. So instead of coming in here for 5 hours with RPTs to do all the measurements, we can do it in like 1 hour, right? So it's now evolution to really refine what we learned in Resolve. This study is going to be even bigger. And again, these are the sites in the US, but now there's sites in Europe as well. It's going to try to get about 450 participants. And go for a lot longer, so about 3 years with like once a year kind of visits. So now this will stretch out that data that we knew, with a lot of different motor outcomes in Resolve, and stretch it out even to multiple years. And these two studies will really become like a core infrastructure as we develop clinical trials for the future. So these are the conclusions. Does anyone have a question? Nothing? All right, so inclusion is pretty simple, right? You have to have FSHD, basically. And there's some parts of this study that we're going to try out, like assessments remotely. Like if I had an internet connection to watch you do stuff at home, could we measure that stuff, right? Exclusion is just that you're unwilling to participate. Oh, that's pretty broad, right? And so this is where early onset comes in. So there's no age criteria, and we really need young people on this study. And it's a really big interest because, as Ali mentioned, there's really not that big, wealth of data for younger onset. But FSHD is generally more severe in these younger kids, and we don't really think that you can totally project from the way an older adult progresses down into these younger age groups, right? We don't know that it's really the same kettle of fish at all. So having a pediatric focus study is really important. And in fact, it may inform the rest of the field in ways that you just can't do. And that's because the more rapid progression, we could actually tell if a drug was working faster in a child than we could in an older person who might be progressing much more slowly. And then as it relates to studies that we're doing, like in our lab, we're interested in genetic modifiers, right? So what makes a person more severe? And now we have this more severe group. And so if there's a genetic modifier making a person more severe, we're more likely to find that pediatric group. So we're really interested in identifying younger patients for us, just in our own research too, not just with MOVE. So I think there's a lot of reasons to be really tuned into MOVE. And there may be a few reasons to be like a little lukewarm about it. You're like, well, it's not a drug trial, right? But this is what really sets the stage for a drug trial. And what will set the stage for the FDA to approve a drug. So like what would be the worst thing that happened is if FDA approves a drug next year for people 18 and over with FSHD, and your kid is 15, right? That would be pretty awful. So the more we can get kids' data into these studies, the better. Because we don't want to have them have to wait, right, for some drug that might actually really help them right now. I don't know if the FDA is really apt to do something like that. They've really surprised us in both Duchenne and SMA to approve very broadly for drugs that get approved. So for like SMA, we had the studies done for one of the drugs in particular done in infants, but it was approved, all the way through across the whole lifespan of SMA. So it's hard to predict, but then we run into insurance problems where the insurers say, well, no, we're not going to cover it because it wasn't studied. So I think we just need to get as many kids, and there's a much smaller group So as many pediatrics as we can get involved. And I think Dr. Tawil and Dr. Statland's that really have been the two people coordinating the clinical trials research networks have recognized that. We're actively seeking funding to do really a pediatric specific study. So this would be sort of a sub-study within MOVE that kind of keeps the same architecture of it, but really does a few things focused in on younger people. And I think that can be a really important study as we move forward. And I know that there's a lot of interest, both myself and with the rest of the network, to do that.
Dr. Butterfield, you mentioned something to me at the Connect conference about kids that aren't showing signs yet and why it's important for even those kids to be included. I was wondering if you could kind of just tell everybody what you told us there, and also reiterate that you don't have to have a genetic positive test to get your kid in. You could have a clinical diagnosis, correct?
Right. And so genetic testing it's still really hard to get clinical genetic testing for FSHD. It's getting easier, and there's some sponsored programs now. I think that'll get easier and easier. The criteria, I think, is that you have to have a clinical signs and symptoms of FSHD and a family history. That sort of gives us enough that we need to know that, yeah, you probably have FSHD. You can get on the study. We do Peter Jones's testing as a part of the study. So that'll be just wrapped into it. Some people have done that already, so they kind of already know from their methylation testing that they have FSHD. So that helps as well. But so you don't have to have like a formal like clinical genetic test to get on the study.
But so parents are saying, well, my kid, I know that they have it probably, or maybe they've been diagnosed through Peter Jones, but they're still mobile. Maybe they're showing very.
Like why would we put them on, right? Those people are really important. And they're important in part for our genetic modifier study. And I'll tell you why they're important for that. And for us, we actually like to get both parents and the full set of brothers and sisters for our studies. We think with patients that we're seeing in our very uniform Utah family that all have the same repeat size, that every person who has the mutation has clinical features. They don't always have them when they're young, right? But we think as the repeat sizes get bigger, there may be people who may not have very severe symptoms, or they may never develop symptoms, right? And it's important to know that, to really be definitive about that. But then we have people who are kind of discordant, right? So, and I've got a patient at my clinic, he has 8 repeats, but he's actually early onset. So how is that happening? And is there a second gene? So for our genetic modifier studies, is what is the other gene that's making that worse, right? And to know that, having the sibling who's milder maybe with 8 repeats helps us find that gene. So what's the difference between those 2 siblings, for example, genetically, that might predict why one was more severe than the other? But also, we want the drugs to be approved across the board too. And so having that full set, what is real natural history? What is the progression? We just need to identify them, right? We need to have them on the study. We need to be able to look a little deeper at what are the things we can see, right? Maybe they're mildly affected, but what is their progression? What are the things we can expect to change in a year? And at the end of the day, if we have a treatment that works in FSHD, it will work better the earlier we use it. Right? That just kind of stands to reason. We have to demonstrate that and do the science, but that stands to reason that the earlier we get that therapy onto your muscle, the less progression you have, the more reversible things can be. And so we just need those milder patients, right? We need to know what their progression is like. We need to know what happens to them.
Dr. Butterfield, I had another question about the MOVE study, and this was a lot of chatter on this on Facebook about, well, people are hesitant to put their kids or themselves on the MOVE study because they think that it will disqualify them for a medical treatment study, a drug treatment study.
Yeah, and which is not true, right? That just, you can be on a MOVE study and be on clinical trials. There's really 2 paths for that, right? One, the MOVE study already knows that we don't want to stop you from being on a drug trial, right? So they've written into the structure of the trial that you could be on both. And once you get in MOVE, it's not all that frequent of assessments. So I think it's like feasible for people to be on both. And we really even want to keep capturing the data in this longitudinal period, right? So we want to make sure that's available. Sometimes the drug trials are going to say no. They're going to say, no, you can't be on 2 different trials at the same time. And if they did, we would just take you off, right? We wouldn't say, we still want your data. We still think it's important. But if you had to come off, I think you can come off a trial at any time, right? That's a part of consent for a trial, and you could come off. And I think we understand that as sort of a community. We faced this a little bit as we've rolled out Duchenne and SMA things, which are just maybe 5 years ahead of FSHD, right? Now we got so many trials and all these drugs approved and things going really fast. But we faced some of that same thing. Like, okay, do I put someone on a trial? And it gets to be, you have to make decisions about that. But ultimately, you could just come off the MOVE study if it came to that. But we, as a clinical trial research network, really are committed to trying to keep you on both if we can.
Thank you for clarifying that. I appreciate it.
What other questions are there? I had just one other slide actually too, but was there a question too? And this is just trials that we've done here in Utah, just because I want people to know on the early onset side that, wow, there's trials, right? They're happening. They've been going on for a while and they're gaining steam. So 2015, and I was guessing dates in these trials take more than a year, but sort of right around 2015, we worked with a company called aTyr. And they had this drug, it's a tRNA synthetase, kind of an anti-inflammatory idea. That trial was actually kind of focused on young, sort of adolescent, young adults, people with young onset. And that trial kind of fizzled. Right? I don't think aTyr is working in FSHD anymore, but it was an experience with a trial that we learned some things from. It was really important. 2018 or so, we started the Acceleron Trial, and I think people may be familiar with this one more. This is an idea called myostatin inhibition. It would block a hormone that slows muscle growth. And we were directly injecting that into muscles. And that actually worked. Actually, the muscles got bigger, but they didn't get stronger. So I think Acceleron has kind of dropped out from that too. So that's 2 trials that we've done that didn't seem to be doing very much. And now this 3rd trial, and I think people have been paying pretty close attention to this in the community from Fulcrum. This drug's a P38 inhibitor. This is one of those pathways that, remember, DUX4 turns on other genes. This is one of the other sort of genes and metabolic pathways that it turns on. And they just finished their phase 2 trial, and they actually had this interesting failure to what they were looking for is the DUX4 turns on other genes, and so they were looking for the levels of those genes to see if we stopped them from getting turned on. And they really didn't do a very good job. They actually couldn't show that. But they could show that one of their motor function tools showed a difference between patients that were treated or patients on placebo. That's called the reachable workspace. And that's one of those tools we've been working on in Resolve. And I think we'll be working on it in MOVE as well. And so now they're starting a phase 3. I think they enrolled their very first patient this month in the phase 3 trial. And so that's moving forward, right? This is now a trial, probably the first one I'm aware of that went to phase 3 in FSHD. And now I'm talking to these other 2 companies, Dyne and Avidity, that are really just getting started at sometime in the next year. And these are getting a little closer to things I think of as gene therapy. So these are using a mechanism to knock down DUX4. And remember I talked about that idea of the way the information flow, the central dogma, and we could block things at different levels. This gets at ways to block that DUX4 RNA from getting made and that gene getting turned on. And those are going to start in the next year. And hopefully you can see that like these are coming. There's more and more trials coming, and we need early onset people to be a part of those trials right now, right? And they're just not quite getting written into all of them in the way that some of them will sneak down to age 16. And there's some regulatory reasons for that. So the way that the FDA is sort of built around things that affect both adults and kids, they'll insist a little bit that they'd be done first in adults. But I think the patient community can have a voice in that, right? And say, no, we need these at younger groups. And there's some scientific reasons, I think, why that would be a good idea, like to get into these younger, more fast onset.
So Dr. Butterfield, how does the patient community, how do we advocate for our kids? What's the best way to advocate for our kids to get them in these trials? Because like you said, the earlier they start taking the drugs, the better. Like if I could get my kid in a trial today, I'd put him in, but we can't. So what can we do?
I think this gets down to trying to communicate well with the FDA that we need this. Because the way that these companies get advice is they go to the FDA, right? And they ask the FDA, they show the data, right, from their animal model or whatever. And they say, we want to get permission to do a trial in humans. And the FDA is going to give them some guidance on that. And there's a regulatory framework around that that's a little bit restrictive for kids. One is we can just show it has a big impact on pediatric patients, right? So actually doing things like MOVE, where we can show the impact on kids, makes them more interested in letting us try it in kids, right? And really, I don't know that that's well understood at the FDA. So just telling that story.
So I specifically spoke to Dine and Avidity about getting kids in trials. And what they kept telling me was, we need more natural history. Yeah. We need exactly what you said. We need to show that it's more severe. Because I can tell you what, as a parent and probably I'm not an FDA FSH patient, but I know FSH patients, they're frustrated. And I'm frustrated as a parent because a lot of the literature on FSHD is like, oh, it's a slow progressing disease, and you might not be able to smile or lift your arms, but it's not going to affect your life all that much, which is very frustrating for all of us, especially the early onset. Well, for me, because that's just my experience, when they're so severely affected. And so they're just like, well, there's no natural history. You can sit here and tell me all day long, or I can look at your kid and say that they're severely affected, but the FDA needs natural history to say proof that look at how severe these kids are, or other FSH patients.
Yep, we just have to do that work, right? We have to just tell that story. And then FDA will do meetings with patient groups, right? And when those meetings happen, and one thing that's really new over maybe the last decade at FDA is really trying to be more tuned into the patient voice about things, even specific things in clinical trials, like what do patients care about, right? Like I care about things when I design a trial, like motor function outcomes, but maybe families and patients care about different aspects of what a treatment would look like. What would a treatment success look like, right? Which symptom, if we treated it, would be most important, for example. And being at the table when those conversations are happening with FDA is really important, because then your voice is heard there, right? And then when they give advice to these companies, that's the advice that they get, right? That stories in their mind of the kid who had onset at age 5, right, and has severe scoliosis, or you know how these things progress. It's not like the same as an adult.
So what the early onset chapter, what we're advocating for, a few different things. First, get your child registered in the U of R registry, so that they're visible in the community, and so their numbers are counted. To get involved in the MOVE study so that they have some natural history, and so that farmers can go back to the FDA or the powers that be and say, this is how severe it is and this is how it affects their life. And then also get involved in like the true cost of the FSHD study, so that if and when the treatment is available, insurance companies will be more inclined to pay for it, because over the lifespan, if it costs $1 million, for this kid over a lifespan, but the treatment is $300,000, then they're more likely to pay for the treatment. So those, and we're also trying to document with video. Kristen in Massachusetts, she's starting to document her child's progression, and we want to get more children documented so that we can do what you were just saying, get their faces in front of the FDA and their severity noted by the FDA and the pharma, so that they'll take note and say, yes, this is a life-altering disease at an early age. Is there anything else that you can think of that we could be doing?
Maybe reiterate the point about getting out and visible and knowing the numbers, right? So a company that sees more numbers will be more likely to go there, right? They're going to flow right to it, to the higher numbers. So the more we have it out there, the more that happens. And just to say, I put up, if you're interested in MOVE, Michaela Walker is the coordinator for that trial, like nationally. This is her email and phone. So, you know, if you're here in Utah, Sarah's on the call, and this is her email and phone. You can just call Sarah, or if you're interested in our genetic research, you can call her too, or just this, I put this QR code that goes right to us. And then we know you, right? So us knowing you helps with that too, because then we can do some of the genetic stuff. But also that puts you in our radar too when we start thinking about like a pediatric MOVE study. And most likely that won't be all the centers, right? That'll be probably a smaller number of centers that are more pediatric focused. We'll have people travel and really build that expertise among a smaller group. But there'll be MOVE centers you can enroll right now. Most of those centers are open to enrollment, and you saw the map there. So, but Michaela can help you get connected to someone close to you.
And something along the line too, so if you're interested in research, but you're not ready to commit, it's still okay to reach out to us and ask questions. Sometimes I even tell people, if you just want to check in to see what's the climate of research or what's going on, definitely please feel free to email me, call me, and I'm happy to talk to you about those things as well. So contacting us doesn't like sign you up for anything. You know, you don't have to be forced to participate or anything like that. So even if you're just curious and you want to chat, like definitely reach out to me.
I've asked a lot of questions. Are there any other questions from any of the parents or anyone on this meeting before we wrap it up? We've gone just over an hour.
I just have one here in the chat that I'm reading because it's kind of long. Someone who had to drop off that has a question about the most promising therapies being developed, CRISPR or Fulcrum. We kind of maybe address some of that. I'd love to know how realistic it is to expect therapies available for children in the next few years. So some of these things I think we've talked about. Maybe just to put a cap on that question, I think there's a lot to hope for in these newer therapies. I think there's really a lot to learn about FSHD, about the genetics and the mechanisms of how FSHD works. Is such a unique genetic mechanism, right? And this idea of the genetics and the epigenetics, how to control methylation and do all these things. Some of these things, you know, the losapramid is a pretty interesting study, and it had an interesting little bit of a signal. It's not the game changer that we want. But it's really a step. Every one of these trials, even the ones I went through that, you know, maybe didn't get us there, we learned something from every one of those trials. And we learned about the natural history. We learned about the motor function tools we were using, and did they work well or not work well, or kind of how did they respond in studying the clinical trials. They all really build. And just to maybe share sort of my experience with Duchenne, like sort of getting into that after I finished my fellowship, is more than a decade ago now, not that much, but it seems like a long time ago. When we first started working in Duchenne, we were taking every study we could get, right? Like 100%. And I had a partner here, Kathy Spoda, who was taking every SMA study she could get, even that weren't that well promising. And now, 12 years later, I have 4 different exon skipping therapies approved for Duchenne. I have a full-out gene therapy approved for SMA, as well as 2 other like genetic manipulation therapies approved for SMA. They all work amazingly well because SMA is easier to treat. But that all happened in less than 10 years, right? And now with SMA and Duchenne both, I turn away really good studies because I don't have the bandwidth to do them. there's so many things happening all at the same time. And FSHD, we're kind of on the cusp of that. And I think I tried to sort of show that for the trials, just the ones we're doing here. So we've got Dyne and Avidity and now Epic getting into the game. We have so many people and companies interested in FSHD. We're gonna get there, right? And I think every next one seems better than the last one. So I really like Dyne and Avidity's ideas about how to knock down DUX4. I think Epic's idea of adding methylation is pretty interesting. It gives me a pause about like whether that's safe or not, but that's moving forward, right? People are using all these new genetic tools and these CRISPR tools and other things to really apply them in ways that I think will have a lot of impact down the road. And it'll be harder not to crack than SMA, but that's okay. That's just, it's an engineering problem, right? We'll figure it out. So anyway, that's the answer to that sort of longer question that was on the chat.
I don't have any questions, Dr. Butterfield. I just wanted to tell you thank you for spending your evening talking with us and sharing your knowledge.
Oh, it's good to be with you. Hopefully we can stay connected as we go to the conferences and do things too.
Perfect. See you all next month.
Thank you so much.
Yes, thanks for coordinating it all.
Welcome. Well, thank you, Dr. Butterfield. If we don't have any other questions, thank you again for your time. And Sarah, thank you, and all the parents that joined. This will be recorded, and if I get any emails, you can email me at Early-onset Chapter at FSHDsociety.org, and I will forward them to Sarah or Dr. Butterfield, and he can answer them. But thank you so much. I appreciate your time. Thank you for explaining, severity and the importance of the MOVE study. And thanks for being a voice for the early onset patient. I appreciate it.
Thank you. Have a good night, everyone.
All right. Good night, everybody.
Thank you.
Thank you so much.